So we are talking about the eukaryotic cells, in which we have discussed about the cell membrane, cell wall. Now we will talk about the endomembrane system. What is this endomembrane system? The endomembrane system in the organelles in these are like coordinated in their function. That's why it is specifically called as an endomembrane system. And endoplasmic reticulum, Golgi complex, lysosome and vacuoles. These are the 4 organelles inside this system. Now we will talk about each in detail. So talk about the endoplasmic reticulum. These are the network of tiny tubular structure found in the cytoplasm. Okay, and these divides the intracellular space. Intra means inside, so cellular means intracellular space. Divide the intracellular space into 2, which is luminal and extraluminal. Luminal means inside ER. Whatever the space inside ER is called as the luminal, and what are the space covered in the cytoplasm are called as the extraluminal. And these endoplasmic reticulum are of 2 types, which is rough endoplasmic reticulum and smooth endoplasmic reticulum. In terms of rough endoplasmic reticulum, it is characterized by the presence of ribosomes, small dot structure if you see in the photo. So, and it is the place of protein synthesis and secretion, because we already, in talking about the ribosome, we said that the ribosomes are the site of protein synthesis. So here we have ribosomes, so it means rough endoplasmic reticulum synthesis the protein and also secretion. Now it has connected with the outer membrane of nucleus. What are the characteristics of rough ER? Presence of ribosome, protein synthesis and secretion connected with outer membrane of nucleus. We will discuss about this later while we discuss about the nucleus. Now to talk about the smooth endoplasmic reticulum, there is no ribosome and it helps in lipid synthesis, and in case of animals it synthesizes some steroidal hormone which is a lipid like. Okay, these are the 2 types of endoplasmic reticulum. Now we will talk about the Golgi apparatus. This is named after the Camillo Golgi because he is the one who found this or like observed these material. These are like generally densely strained reticular structure. Okay, these are like densely strained, but in case of endoplasm these are like tiny tubular structure. So these are the difference about each organelle in this shape. And these Golgi bodies contain cisternae. These are nothing but some flat or disc shaped sacs. Okay, flat or disc shaped sacs, or it is called as cisternae, and these are of 0.5 microns or micrometers to 1 to micron. So 0.5 to 1, this is the size of the cisternae. These are arranged especially near the nucleus with either convex or cis face, concave or trans face. Okay, it has 2 faces, one is convex and convex, and near the nucleus it may be attached either with this convex or cis face or concave or trans face. The convex is a forming phase and concave is the maturing phase. These both are entirely different but they are interconnected. Okay, this convex and concave phase are entirely different but they are interconnected. These Golgi bodies perform the packaging of material. The main function of the Golgi body is to package the materials. This packed materials will either sent to the intracellular within the cell or to the outside cytoplasm or any other organelles. The material to be packed in the form of vesicles from the endoplasmic reticulum will be fusing with the cis space. You are getting the point. Okay, so the materials which are need to be formed in the form of packed in the form of vesicle will be joined at the endoplasmic reticulum, and it fuse with the cis phase of the Golgi apparatus and it move to the trans phase. And this becomes the reason why the Golgi body and the ER, that is endoplasmic reticulum, are close in structure. Now let's see how this is happening. Firstly, this proteins comes from the endoplasmic reticulum will be modified from the cisternae. Then it will be do the packaging things. Then it will be released from the trans phase. This is how the packaging of materials takes place in the Golgi body. Now we will talk about lysosome. So these lysosomes are formed when the packaging process is happening in Golgi apparatus. We talked, right? The Golgi apparatus is mainly for packaging of material. So whenever this is happening, during that process, lysosomes are formed. These lysosome vesicles are high in hydrolytic enzyme. Okay, they contain more hydrolytic enzyme and hydrolysis such as lipases, proteases, and carbohydrates. Hydrases. As you all know, these are useful for the digestion of the specific enzymes. In case of lipases, it will digest the lipase. Okay, so then it will activate the acidic pH. It has the ability to activate the acidic pH. These are all about lysosome. Now we will talk about the vacuoles. The vacuoles are nothing but the membrane bond space. Okay, these are some space which are found in the cytoplasm. So what are the materials found in the vacuoles? It's a simple space, right? So it contains water, sap, excretory products, and also not useful product for the cell. And these are single membrane. Okay, these are made up of single membrane. Whether the organelle is made up of single membrane or double membrane, how to memorize it, I have given a trick video for that. You can go and watch it. So it will be easier for you to, instead of memorizing each and every one. Now this single membrane, it is called as tonoplast. In case of plants, 90% of the volume is occupied by this vacuole only. So it will be in large in size. Now the concentration in the vacuole is higher than in the cytoplasm. It's because in the vacuole it will be the transport facilities, it will be in the against the gradient. What is against gradient? It means like it moves the low to higher, that is against the gradient. That's why the concentration in vacuole will be more in the it is more than the cytoplasm. And to talk about the different type of vacuoles, in case of amoeba we have contractile vacuole which help in excretion. In case of protist we have food vacuole which helps in engulfing the food. It will just engulf the nearby food. Okay, here we have discussed about endomembrane system and ER, Golgi apparatus, lysosome and vacuoles. We will see the questions for this. The first question, what are the organelles found in endomembrane system? Which is the site of lipid synthesis? Give the size of cisternae found in Golgi apparatus. Convex phase is called as forming or mature phase. What is the membrane of vacuole called as? What is the percentage of vacuole in plants? Food vacuoles are found in. I hope you have able to answer all these questions. If not, revise it once along with the NCERT. Then we will move to the next page. So lastly, we have discussed about the endomembrane system and the organelles inside it in detail. Now we will talk about the mitochondria. This mitochondria will be different in number and size in each of the cell depending upon the need, and these are double membrane. So I have made a trick video as I said before, so to memorize what are the organelles which are of double membrane and single membrane and membraneless organelles. Okay, so please do watch that video. Now we will talk about the mitochondria in further. So this in double membrane, one of which is inner and other is outer. This inner membrane will divide the lumen of the mitochondria into two compartments furtherly. One is outer compartment and inner compartment. Okay, so there are two membranes which is inner and outer. This inner membrane will divide the lumen into outer component and outer compartment and inner compartment. This inner compartment is called as the matrix. Now this outer compartment will form the limiting boundary of the organelle, but the inner compartment forms the number of infolding which is called as cristae. In case of Golgi apparatus we talked about cristae, but here it is cristae. Okay, so outer compartment forms the limiting boundary like stopping, but the inner compartment forms the inner folding. It is called as the cristae and it helps in increasing the surface area. And these are towards the matrix. Matrix are nothing but the inner compartment, right? Now, these mitochondria are the powerhouse of the cell because they has the aerobic respiration site. They produce ATP, that is energy. That's why it is called as the powerhouse of the cell. Apart from this, we have some things in the matrix which are nothing but the single circular DNA, few RNA, and 70S ribosomes, and also synthesis of proteins is also taken in the mitochondria. These are the functions and characteristics of mitochondria. Now to talk about plastids. Plastids are found in plant cells and in euglenoids. Based on the types of the pigments, there are 3 types. One is chloroplast, next is chromo, and another is leucoplast. These chloroplasts are containing the chlorophyll, that's why it's named as chloroplast, and carotenoid pigments, and these are help in photosynthesis. Obviously, whenever you say chlorophyll, you should remind immediately about photosynthesis. Now to talk about the chromoplast, it has the fat soluble pigment, carotenoid pigments such as carotene, xanthophylls, and it gives the color to the plant such as yellow, orange, and red. Now to talk about the leucoplast, it is colorless. In case of leucoplast, it is colorless and it stores nutrients in 3 types. One is amyloplast, eleoplast, and alluroplast. This is in the form of storing the food. The amyloplast will store carbohydrates, which is starch, and eleopols, like as the name indicates, it stores oil and also fat, and alluroplast stores protein. These are about the types of plastid. And to talk about the chloroplast, these chloroplasts, as discussed, it contain chlorophyll, and these are generally found more in the mesophile region. These mesophiles are nothing but lens or oval or spherical or ribbon shaped structure found in the plants, and these mesophiles are differ in numbers. In case of chlamydomonas, it may be 1, but in case of green algae, it may be 20 to 40. Now, these are contain double membrane, and I have trick video for that, in which the inner membrane, the space limited by the inner membrane, is called as stroma. This stroma contains flat sacs. These are called as thylakoids. Okay, the place limited by the inner membrane in the chloroplast is called as stroma. This stroma contains flask or flat sacs, which are called as the thylakoids. These thylakoids will be stacked or like arranged in the like a piles of coins, and that is known as the grana or intergranal thylakoids. So thylakoids together form as a stack, which is known as the grana. Okay, so between thylakoids of different grana. Okay, what are the thylakoids? Thylakoids are single structure, single flat structure. Grana is when thylakoids are arranged together. So between thylakoids of different grana, there will be stroma lamellae. Say this is one grana that is arranged in like a pile of coins. This is for example I have drawn like that, and this is another grana. So between the thylakoids is these single. So between the thylakoids of the different grana, there will be a stroma lamellae. This is what it detects. And also in thylakoids we have chlorophyll pigments. Okay, so the chlorophyll pigments are present in the thylakoids of the chloroplast. To continue with the chloroplast, we have another structure called lumen within that, which is nothing but the space which is enclosed by the membrane of thylakoid. The space which is enclosed by the membrane of thylakoid is called as lumen. This stroma. What is stroma? Try to revise. It is a space which is limited by the inner membrane of the chloroplast. Right. Inside the stroma, we have synthesis of carbohydrates and protein, that is enzyme required for the synthesis of carbohydrates and protein, small double-stranded DNA, and also ribosome. These ribosomes is of 2 types here. One is that is 70S, which is found in chloroplast, and another is 80S, which is found in the ribosomes. Are we clear with this plastids? Now we will see the questions, but we will complete these also together, and we will see the questions for the previous page and also for this page. Now to talk about the ribosomes, these are firstly discovered by George Palade in the year 1953. Okay, right. They are composed of RNA, which is ribonucleic acid, and proteins, and are not surrounded by any membrane, membraneless organelles. Okay, and in case of eukaryotic, we have 80S ribosomes, but in case of prokaryotic, we talked about 70S. What is this? S denotes Sudbeck's units. Okay. Now we will talk about cytoskeleton. These cytoskeletons are nothing but the filamentous proteinaceous structure, networks of filamentous proteinaceous structure found in the cytoplasm. So these together called as the cytoskeleton. They has the mechanical support, motility, and also maintain shape. Now we have cilia and flagella. Cilia and flagella are the locomotory organs, we can say, and these can be small. I mean, cilia are the small one, flagella are the large one. These are nothing but the hair-like, like a minute hair-like soft projection outgrowth. These are covered with the membrane, and we have axonum, which is nothing but the core part. The core part of the cilium or flagellum is called as the axonum, and it has 9+2 array. So what is this 9+2 array? We have 9 doublets which are arranged radially. Okay, radially peripheral arrays, but in central we have 2. So 9+2, 9 outside and 2 in central. That is called as the 9+2 array. These in central we have 2, right? These are connected by bridges, and the central tubules are covered by central sheath. These are connected by bridges and covered by central sheath, and these central sheath will be attached to the one of the peripheral tubules. It will be in doublets, right? It will be in doublets. It attached to one of the peripheral tubules like this. 2 are there. It will attach to one in the terms of radial spoke. Okay. The central sheath, which is surrounded by the central tubules, are attached to the peripheral tubules in terms of radial spoke. And these peripheral tubules too are connected by the linkers. Okay. I'm sorry for confusing you. These peripheral tubules are connected by linkers, but the central tubules are connected by the bridges. Central and peripherals are connected by radial spoke. I hope you are clear now. And where are these cilia and flagella coming from? These are emerging from something called as basal bodies, and these basal bodies has centriole-like structure. Now we will talk about the centriole. Centrosome and centriole. The centrosome is an organelle which contains the centrioles. These centrioles are cylindrical in structure, and in centrosome we have 2 cylindrical structures, that is centriole, and they are arranged perpendicularly. As like in cilia and flagella, here also we have 9 peripheral fibrates, but these are in triplets. In case of that we talked about doublet, here it is triplet, and these are nothing but the tubulin proteins. And the central part of this is called as the hub. Okay, this hub is also made up of proteinaceous layer, and this hub and tubulin proteins, that is peripheral fibers, are attached by radial spoke. This is about the centriole and the centrosome. Now we will see the questions for the previous and this page. The first question will be, what is cristae? The next, which is called as a powerhouse of the cell. Ribosome in mitochondria are of 70S or 80S. Allotrope stores what kind of materials? Chromoplast contains which pigments? What is grana? Ribosome in eukaryotes 80S or 70S. How are the peripheral tubes or interconnected? Central proteinaceous part in centriole is called as. I am not discussing the answer because just now we are revising the concept, so I thought you will be able to find the answer also. Now we will move on to the next page.